Hi everyone, I am Isabella Saldana,
I'm August Brookwell.
I'm Eli Balderas.
and today Eli, August, and I are going to take you through the process of RNA interference and RNAi knockdown. Let's get started. So what is RNA interference? RNA interference, or RNAi, is a process by which double-stranded RNA induces or directs gene silencing by targeting mRNA and degrading it. It is a highly integrated process throughout cell biology and is used by eukaryotic cells as both a regulator of translation and a defense mechanism. Additionally, RNAi knockdown can be used in the lab as a powerful tool for post-transcriptional gene silencing. It has been used to study the cellular functions of particular genes by inducing silencing or loss of function genotypes. It's also been used to study a number of diseases, including cancers, HIV, and hepatitis. In crop science, it has been used to provide resistance against parasites in certain crops as well. Now we'll take a closer look at the mechanisms of RNA interference. There are two primary pathways involved in RNAi, both of which involve small RNA molecules, one of which being microRNA, and the other being short interfering or siRNA. While the processes are fundamentally similar, we'll be focusing a bit more on the rightmost pathway, involving siRNAs. So the microRNA pathway has an additional transcription and processing step in the nucleus, where the precursors to microRNAs are processed before exiting the nucleus via an exportin protein and moving into the cytoplasm as double-stranded RNA. The presence of this double-stranded RNA in the cytoplasm attracts the endoribonuclease dicer, which cuts the double-stranded RNA into fragments about 20 nucleotides long. These fragments bind Argonaute proteins, abbreviated AGO, after which the double-stranded RNA dissociates into two strands. The antisense strand, also called the guide, stays bound to the Argonaute protein, while the other strand, called the passenger, falls away and is degraded. Together, the Argonaute proteins and the guide strand are collectively called the RNA-induced silencing complex, or the RISC, as you can see in both pathways here. Guided by the antisense strand, the RISC binds to the target mRNA strand via complementary base pairing. Binding may be either complete or incomplete. If the complementary binding between the antisense strand and the mRNA is incomplete, then translation is immediately repressed and the mRNA is eventually degraded, as we can see here. If the binding is exact, as is the case for many plants, the mRNA is immediately cleaved by the Argonaute and degraded. As mentioned before, this mechanism offers protection to the cell and is especially effective against RNA viruses in eukaryotic cells because it can recognize the insertion of viral double-stranded RNA into the cytoplasm and efficiently target and degrade it. Plants have an additional level of protection in that they can spread RNA interference activity between their cells, which gives the whole organism immunity to foreign attackers, even if only a small number of cells were affected. Researchers have taken advantage of the RNAi mechanism to study post-transcriptional gene silencing for many applications, and this method is performed in several steps. First, the process requires a gene-specific siRNA for the gene of interest. This is usually done by preparing the siRNA directly in vitro, which is generally done through chemical synthesis. In vitro transcription, digestion of double-stranded RNA by endoribonucleases, the use of plasmids and viral vectors may also be used to introduce siRNA into cells. Next, you need a method to transfect this siRNA to the cell. Lipid or amine-based reagents are the most common, but electroporation might also be used depending on the cell type. After transfection, you need a way to assess the effect of the RNA knockdown. qPCR and Western blotting are commonly used assays for assessing target transcript levels and target protein expression, respectively. Finally, you would need proper controls, the most common of which are validated non-silencing siRNA, which we'll see in our example later on. Now, here's August with a helpful analogy for thinking about the process of RNAi.
So this is our RNAi analogy on baking cookies. So a chef, which represents the ribosome, and his team of cooks, which represent transcriptional and translational machinery, wants to bake a batch of cookies, which represents the protein. So one of the cooks looks up a recipe online, which is representing the DNA, and writes a copy of the recipe, which is representing the mRNA, to bring to the chef. So before the recipe can get to the chef, however, it gets lost in a pile of other recipes, which just represents that mRNA sequence being in a greater mRNA transcript. So the sous chef, which is representing Dicer, thinks this is not a good recipe and does not want it to get to the chef. So to find the original recipe in the pile, the sous chef, Dicer, cuts the identical recipe, which is the foreign dsRNA, which is cut into either mi or siRNA, from his cookbook, and hands the recipe to the cook, which is representing risk, to match with the original recipe. So the cook matches the two recipes and cuts them. And the result is that the chef never receives the copies and the cookies are never made. So translation never occurs. This is the paper that we went through. It's titled RNAi knockdown of PAR4 inhibits neurosynaptic degeneration in ALS-linked mice. So amyotrophic lateral sclerosis, or ALS, is a well-characterized neurodegenerative disease. So characteristics of ALS include a loss of motor neurons in the spinal cord and the brainstem, as well as paralysis in patients. Cellular apoptotic cascades have been suggested to be linked to the degeneration of motor neurons observed in ALS patients. And prostate apoptosis response 4, or PAR4, has recently been observed to be linked with this degeneration. PAR4 is a protein. It consists of death and leucine zipper domains, and it mediates neuronal degeneration. So it experiences increased levels of expression in neurons undergoing apoptosis. And PAR4 has also been observed to be in much higher concentrations in the spinal cord samples of ALS patients as compared to those without ALS. So in mouse studies with ALS, PAR4 was observed to play key roles in motor neuron degeneration. The underlying mechanism by which PAR4 is linked to motor neuron degeneration is unknown. However, it is thought that PAR4 might be involved in disjunction of synaptic contacts that connect the cerebral cortex to the spinal cord and brainstem. So dysfunction in these contacts is a hallmark of neurodegenerative diseases like ALS. Separate from that, ALS can be brought on by mutations in SOD1. Specifically SOD1 containing an amino acid substitution of glycine to alanine at position 93. So in mice, this mutation increases PAR4 expression in the synaptosomes. And using RNA interference targeting PAR4 expression, spinal motor neurons were protected against apoptosis in mice. So this suggests that PAR4 expression is linked to ALS.
So to test whether RNAi knockdown of PAR4 protein expression could rescue spinal synaptosomes and motor neurons from apoptotic damage, transgenic mice with various stages of ALS symptoms were transfected with short interfering RNAs that targeted the PAR4 protein. These mice had a G93A mutation of their SOD1 gene, causing enhanced PAR4-mediated apoptosis of motor neurons. And the variable levels of PAR4 expression in the mice was analyzed using Western blot analysis, which is a technique used to separate and identify proteins through gel electrophoresis. So short interfering RNAs were generated by in vitro transcription, in which T7 promoter sequences containing nucleotides from the PAR4 encoding sequences were added to a DNA template. A transcription reaction was then assembled, forming complementary RNA, and the RNA samples were then separated from the DNA template and double-stranded RNA was purified. Lastly, the short interfering RNAs were then fluorescently labeled and transfected into the primary spinal motor neurons, and this allowed researchers to determine the efficiency of transfection into the cells. And so in figure B, we can see that the fluorescently labeled short interfering RNAs are showing up in the motor neuron cells as the bright red spots, and this means that the transfection was successful. And comparison of PAR4 expression levels in wild type and transgenic mice confirm that PAR4 expression is increased in transgenic mice with ALS symptoms, and that RNAi knockdown of this protein could protect the mice from motor neuron degeneration. This was determined after confirming several results in combination. And so the first one was that RNAi knockdown of PAR4 expression was successful, and that's evident from the Western blot analysis in figure A, in which the PAR4 proteins are drastically reduced, while control proteins were still expressed. So additionally, the specificity of the RNAi knockdown was proven to be accurate by comparing knockdown of PAR4 with control groups of non-silencing RNA, and by testing the effect of the short interfering RNA on other proteins. So in figure C, you can see that the PAR4 expression, which is induced by glutamate, is knocked down by the short interfering RNA, but is still showing up in the fluorescent microscopy of the other control groups. And in figure F, presenilin 1 is the protein showing up under fluorescent microscopy. So for the motor neurons in which short interfering RNAs against PAR4 were transfected, the presenilin 1 protein was still expressed, meaning that the short interfering RNA specifically targeted the PAR4 protein and didn't interfere with other protein expression. And these results in combination support the findings that RNAi knockdown of PAR4 inhibits neurosynaptic degeneration in ALS-linked mice. Thanks for watching.